Hello, everybody. Good morning, good afternoon, and good evening from wherever you are logged in from. And a warm welcome to this webinar on viral vectors in vaccine development. This webinar is the eighth and the final one for this year that we're carrying out as a series of planned webinars with the goal of providing information on available career options in the field of vaccinology. My name is Caroline Mata, the Vaccinology Workforce Development Project Strategy and Planning Manager at the KEMRI-Wellcome Trust Research Program, and together with my colleagues, Professor George Warimwe, Marianne Munene, Lavender Nafula, Linnea Imbai, I welcome you to this session. Before we start, would have a few housekeeping matters. Kindly take note that this webinar is being recorded. Kindly feel free to use the chat option to share your questions during the presentation, and we will address them during the Q&A session at the end of the presentation. We will have a few open slots for people to directly ask questions to the presenter. And if your internet is stable, you can raise your hand using the available icon, and I will be able to unmute you. As earlier mentioned, this is a series of planned webinars with the goal of providing information on available career options in the field of vaccinology. This is run under the KEMRI-Wellcome Trust Research Program, which is a world-renowned health research unit of excellence based in Kilifi and in Nairobi in Kenya, as well as Mbale in Uganda. Over the years, we have excelled in the use of novel ideas and collaborations that achieve better health for Africa while also developing the capacity of African scientists and leaders. Our vision at the program is to undertake cutting-edge and novel research relevant to national, regional, and global needs. It is under this program that we have the Vaccinology Workforce Development Program that aims to expand the technical know-how and capacity of sustainable end-to-end vaccine research and development in Africa through training and knowledge exchange activities such as this within the vaccine ecosystem. And this involves areas such as target product profiling, research and discovery, preclinical and clinical trials, biomanufacturing, regulatory affairs, and deployment. We do this through three particular pathways. One, through internships at human vaccine manufacturing institutions that aim to establish a mentorship scheme that links interns with senior vaccinologists and health and market systems while engaging academics globally to share experiences and research skills in the vaccine research and development so that they can enhance their particular career development. The second pathway is through a fully funded 2-week master's level course in vaccinology that is presented both in English and in French to successful applicants who are then selected based on their direct involvement with vaccine research and their excellence in professional and academic achievements. And the third pathway is through webinars such as this one today that targets early career scientists and other scientists, graduate and undergraduate university students in Africa to be able to provide information on potential career options and being able to give them an interactive session where they can be able to get information. So this program is particularly aligned with the Africa Union Partnership for Vaccine Manufacture Ambition, which is to increase the vaccine productivity to meet 60% of Africans' routine immunization needs by the year 2040, and to strengthen the capacity of vaccine research, discovery, and manufacture within the continent. This webinar is part of that engagement. So today we have the pleasure of hosting Christine Dahlke, who will be discussing viral vectors in vaccine development. Christine is a virologist who specializes in immunology, vaccinology, and molecular biology with 10 years of experience in clinical research in an academic environment. She holds a PhD in biology with a focus on virology and molecular biology. She did her PhD thesis on the herpes virus encoded microRNA at the, you will be able to pronounce that for me, institution in Hamburg, Germany. And in her postdoctoral period at the University Medical Center, She combined molecular biology, immunology, and vaccinology and studied innate and adaptive immunity in humans after vaccinations to improve the understanding of how vaccines work and to find markers that predict protection against diseases or infection. Between the years of 2017 and 2020, Christine was not only a postdoctoral fellow, but also the project manager of the section of emerging infections, vaccine research at the UKE. And she has also led the immune monitoring strategy for several clinical trials, and she is interested in development of vaccines against emerging viruses and the evaluation of biomarkers, focusing on viral vector vaccine candidates. After 10 years of vaccine research, at the UKE, she transitioned to a new role as a translational immunology lead at CEPI, and here she developed strategy for the evaluation of immune markers for vaccines, and is a project lead for the Controlled Human Challenge. Christine has co-authored around 35 peer-reviewed articles in infectious disease, clinical research, and vaccinology. She's also a lecturer in molecular biology and is part of the German Society of Virology. I'm handing over to you, Christine. Take it away.
Thank you, Caroline. Thank you, Caroline, for the kind invitation. And I'm honored to be here and talking about the viral vectors and in vaccine development. And I will share my slides. Let's see. So I hope you can see my slides, okay? And today I'm going to talk about viral vectors, and I was not really sure about the audience, so this will be really a high-level discussion on viral vectors. And if you're interested in more deep dive into immune responses to viral vectors or other perspectives on viral vectors, you can always reach out to me later. So my talk is going to be in 4 parts. So at first I would like to highlight emerging infections, then the Ebola outbreak in 2014, the viral vector vaccines, and then giving a brief overview about our 100 days mission from CEPI. So with this slide, I would like to highlight that infectious diseases still pose one of the greatest threats to public health. And this was also experienced in the last 5 years, where we experienced the COVID, Marburg, Ebola outbreaks, and Zika, for example. And the emerging viral infections are the origin most of epidemics and pandemics. And so this 21st century has witnessed a wave of severe infectious disease outbreaks in the past, and the emerging pathogens really continue to pose a serious threat to global health. And the question is how infections really emerge. And one of the important factors that lead to emerging infections are zoonotic diseases. So about 60% of emerging infections are derived from the animal sources. And in general, there are 2 important steps to establish an outbreak. On the one side, you need to have an introduction of an agent to a new host population, which means, for example, you have a virus and in a new region where the population has not seen or faced to the virus before, and then the agent has the first cases. And if you have then an establishment and the transmission within the new population, you will have an outbreak. There are several contributing factors to emergence of infectious diseases, and I would like to highlight 4 of them. And on the one side, you have the climate change, which means that the habitats of vectors are altered, so like mosquitoes and the ticks coming to malaria and CCHF, for example, inducing the diseases in new regions. The globalization means that there's an increase of movement of people, animals, but also goods, which facilitates pathogen spread. We have the urbanization, which means that We are more and more crowded in the living conditions, and this enhanced the transmission and, of course, the antimicrobial resistance, which leads to treatment challenges. So infections have been emerging for thousands of years as interactions between humans, animals, and their environments have increased and changed. And there are three aspects which triggers also or facilitates emerging infections even more. So the changing in human demographics and behavior, the travel and industry, but also susceptibility to infection when we think about elderly population or children's or immunocompromised individuals, increases the risk of emerging infections and also transmission and the outbreak size. So this graphic shows you the International Air Travel Network in 2020, highlighting that when we think about an outbreak and then transfer it to the more frequent travel, to the globalized trade, but also to the greater interconnectivity between countries, these outbreaks are becoming as inevitable as they remain also unpredictable. And the question is then, how can we be prepared, or how can we be better prepared? And one approach, not the only approach, is to use vaccines. And this is one important countermeasure to limit outbreaks and to protect humans against disease and death. So that vaccines work have been shown also in the previous time, and they are on the, I think most of you know the graphic. On the left side, you can see the pre-vaccine era and the estimated annual morbidity in the US, and on the right side, after vaccine implementation. And the most successful story about to prevent disease is the smallpox, smallpox vaccine, where we successfully eradicated the virus. So this smallpox eradication was the first and so far, unfortunately, the only disease to be permanently eradicated from the world. And so in 1967, the WHO pushes really the eradication efforts, and in 1980, the WHO declared the smallpox eradicated. And this was successful because the smallpox could only survive in humans and not in animals. So we only had one population where we could focus on getting the vaccine and stopping transmission and therefore eradicating the virus. So then I would like to highlight the next outbreak, which the COVID-19 pandemic, where we have on the 31st of December on 2019, the first cases of pneumonia in Wuhan. And this COVID pandemic was really the largest public health crisis the world has faced, and over 750 million confirmed cases have been reported, and 20 million deaths have been reported as well. So when we think about the vaccine, this was really an unprecedented speed, because with the identification of SARS-CoV-2, the causative agent, SARS-CoV-2, then a lot of vaccine developers started to develop vaccine candidates against SARS-CoV-2. So, and the vaccine developer have 8 ways to develop a vaccine against a pathogen. And these are highlighted in this graphic. So we have the virus approach, we have DNA, RNA vaccine candidates, viral vectors, and protein-based vaccines. And these ways can be used to deliver the genetic code. For SARS-CoV-2, was it the spike protein, so the most immunogenic antigen to the human. And then this is a slide from 2022 showing how many vaccine candidates have been developed. So 194 vaccine candidates against SARS-CoV-2 have been developed in the preclinical stage. And this means that all the different platforms like the DNA, RNA, vectors, and proteins have been tested. And this was an enormous situation for the vaccine field, because we learned a lot from these clinical trials, from the preclinical trials, from the phase 2, phase 3, phase 4 trials, what kind of platform is useful for SARS-CoV-2 to protect against the disease and against death. In previous time, before the COVID pandemic, the normal time frame was about 5, 10, 15 years to develop a vaccine so that the vaccine is available for use. But in the COVID-19 area, we have really shortened the timeline to 12 to 24 months. And this was kind of really unprecedented. And this graphic shows you the vaccine timelines from other pathogens, and showing that, for example, for polio, we have about 4 years that were used to develop the vaccine for use. And this was also quite fast. But then SARS-CoV-2 showed that we are able to have 326 days to develop a vaccine. And this was based on the prior research on innovative scientific technologies, on funders like CEPI, Wellcome Trust, Bill Gates Foundation, and others, to accelerate the trials to push vaccine development so that we have really a broad vaccine library to be tested and choose then what is the best vaccine for SARS-CoV-2. However, the question now is, after the COVID-19 pandemic, what if we could go even quicker? And this will lead to the question about a 100 days mission, which I will talk later in the talk. So my question now is, how could we develop a COVID-19 vaccine so quickly? And to answer this, I would like to focus on the West African Ebola outbreak, because this was an important area or important period where vaccine development was pushed during an epidemic. And after this, the vaccine development processes and procedures changed, and also the view on how can we be prepared for the next pandemic. So this was kind of a turning point. So the outbreak in 2014-2016 was the most dramatic Ebola virus outbreak in history. And on the right side, you can see the different outbreaks, starting from the first reported outbreak in 1976. In yellow, you have the cases, and in red, you can see the deaths. And you can see that the outbreaks were kind of limited. About 200 humans lost their lives. But then with the West African Ebola outbreak, which hit 3 countries, Sierra Leone, Liberia, and Guinea, this was the most severe outbreak. So more than 28,000 cases have been reported, and about 11,000 humans lost their life. In August 2014, the WHO Director Michael Chan declared the outbreak as a public health emergency of international concern. And then as a response, they said, we want to have vaccine trials. We need to have a vaccine to limit the outbreak. And so vaccine trials were then accelerated. But the problem is now, where are the vaccines for the Ebola at this time point in August 2014? And unfortunately, some of the vaccine candidates were only in the preclinical stage. So these are some of the vaccines that were tested in animals, but only 2 of them, the CHAT and the VSV-EBOV, possess clinical grade 1 material for phase 1 clinical studies. And this is required to move into the phase 1. So only 2 candidates in 2014 were able to enter phase 1 trial. So during the outbreak, there were then phase 1 trials in non-endemic regions, and therefore the affected countries had to wait about 6 to 8 months to get the vaccine to their to the place and to test the vaccine in their affected population. So I was part of the VSV Ebola Consortium WebCon, and maybe some of you were also part of the WebCon, because one site was also in Kilifi. And the WebCon was created to initiate phase 1 studies to facilitate phase 2 and 3 trials in affected countries. And there were 4 parallel harmonized WebCon trials. And one site was in Gabon, Kenya, Germany, and Switzerland. And they tested the VSV-EBOV. And the VSV-EBOV is a viral vector vaccine. It is based on the vesicular stomatitis virus. And this is a live attenuated vaccine, which means that it can replicate in the human body for a few days. So this is the structure how they designed the vaccine. So on the left side, you can see the Ebola virus genome. The genome is quite small, and they used the glycoprotein. So the protein which is on the surface of the Ebola virus was used and introduced into the VSV. And they switched the glycoproteins. So VSV has also a gene for glycoprotein. This was cut it out, and then the Ebola glycoprotein gene was introduced into the genome, in addition to the fact that also the glycoproteins were on the surface expressed. So this was a unique strategy also, that if the vaccine is introduced to the human body recognized the protein on the surface, but then also the gene encodes the glycoprotein. So of course there are some pros and cons, and the con or the drawback of the vaccine is on the one side the safety concern, because it is a replication competent vaccine, which means that the virus replicates in the body, and if you have, for example, immunocompromised patients or individuals, people, they will have maybe a risk after vaccination. It's a new world virus, so you bring a new world virus into an old world virus, into the old world, and you bring a new virus to human because it's the virus for circulating in livestock. The pro is that on the one side, the manufacturing, so it grows to high titers, so it's not super easy to produce. It's not the easiest manufacturing process, but it has a really good production and efficient production. When the vaccine enters the cell, it replicates in the cytoplasm, so there's no risk of integration. You have limited seroprevalence. There's a lack of serious pathogenicity in humans. You have a really rapid and robust immune response, which means also that a single injection, so a single shot, would be enough for protection. And this is super important when you think about an outbreak scenario. So if you are in an outbreak, you want to have a single shot and then you want to be protected. And you don't want to have kind of a complex prime boost regimen where you have different time points maybe on different vectors that need to be administered. And one point is also that it has been reported that the VSV-EBOV is able to protect after the exposure, which is also important for outbreaks, of course. So if you want to deep dive into the VSV story, there are several publications, and I put two reviews here, and just for your interest. So after the phase 1 trial, so we published our data in the New England, but also in the US group, tested the vaccine in a phase 1 trial, showed good immunogenicity and good safety profiles, so we can then enter the phase 2 and phase 3 trial. And the WHO performed the Ebola SARS-CoV trial, which used the ring vaccination. And here they could show that the VSV-EBOV has over 90% of effectiveness. So this was a high efficacy for the vaccine and was licensed after the outbreak. And this is kind of the timeline about the development of the VSV. So in 1996, the VSV was first developed as a recombinant VSV, and 1 year later, there was an introduction of the green fluorescence protein into this VSV genome, And a few years later, Heinz Feldmann and his team developed the VSV-EBOV. And the first trials and first research looked really promising. And so they received good funding from 2001, because also that Ebola was seen as a risk in escalating from localized outbreaks to a global pandemic. And in addition to this, it was classified as a bioterrorism threat. So the US and Canada, they invested into the vaccine. They did non-human primate clinical trials and showed that the vaccine is highly protective in NHPs. And then they entered the manufacturing process, but then stopped the development. So from 2005, there was no investment to test the vaccine in humans. And this was kind of the tragedy when we think about the Ebola outbreak, because in 2014, Ebola hit the 3 countries. And then we didn't have any data on the first phase 1 trial. And we had to take the round of testing the vaccine in non-epidemic regions, in Germany, in Kenya, and in US and Switzerland. And after several months, the vaccine was transferred to the phase 2 trial and the efficacy trial, and then licensed finally. So then the VSV-EBOV was then approved by the EMA and WHO, and it is now a Merck vaccine. And after this approval, another vaccine, the Johnson & Johnson, using 2 viral vectors, the adenoviral vector 26 and MVA, which were genetically modified, they were also approved. They carry the genetic code of several EBOV proteins. So with ending this epidemic, Mimi Dacot highlighted that we should never again experience a crisis like the West Africa Ebola epidemic, and the world needs a more dynamic approach to R&D for life-saving drugs, vaccines, and diagnostics. So the question is now, what have we learned from this tragic experience? And on the one side, there are several lessons learned, but I would like to highlight 3. That viral vector vaccines can be effective in preventing disease and in limiting an outbreak. That we need to better understand how vaccines work, and we need to be better prepared against pandemics. So there is an unknown enemy. So we know that outbreaks of viral infection are certain, so they will occur, but we do not know when and which kind of virus will come. And this was also discussed by Rino Rapulli and his colleagues in a publication from 2017. And here they highlighted that outbreaks remain unpredictable and we have a reactive strategy. So before the Ebola outbreak, there was always a reactive strategy, which means we have a first case, which is shown in the middle. The media is coming, or the press reports about the outbreak. Then the money goes into industry, into biotech, academics. We have a lot of research and develop biologics or vaccines. But then when it's ready to use, there's the end of the epidemic, the end of money, and the end of the project. And so we need to have a different strategy. We need to have a head start, which means before the first case, we need to understand the vaccine candidate, testing them in preclinical studies and clinical studies. We need to have a scale-up manufacturing strategy for this vaccine. But the question is, of course, against what? We can't develop vaccines against all the different pathogens that are out there. And therefore, the WHO developed or created this R&D blueprint team for action to prevent epidemics. And they discussed all different pathogens. And below here, you can see a list of top emerging diseases that are likely to cause major epidemics. And so that vaccine developers and also funders should focus on this kind of priority pathogens to develop vaccines against them, like Ebola, Lassa, MERS, and SARS, and Nipah, and Rift Valley fever. And also Stanley Plotkin and some other colleagues highlighted that we need to change this preparedness for outbreaks. And we need a global R&D funding mechanism aiming to enhance coordination of the R&D process for developing vaccines in outbreak situations. And then CEPI was created. So this is a $1 billion project to prep vaccines before the epidemics hits. And this was launched in Davos in 2017 as a result of the Ebola outbreak, and that everyone had the consensus that a coordinated international and intergovernmental plan was needed to develop and deploy new vaccines to prevent future epidemics. So CEPI is a global coalition to tackle epidemics and pandemics. And on the one side, CEPI has a vision in which epidemics and pandemics are no longer a threat to humanity, and a mission to accelerate the development of vaccines and other biologic countermeasures against epidemic and pandemic threats, so they can be accessible to all people in need. And one approach, so CEPI has different approaches to tackle epidemics and pandemics, but one approach is really to focus on what kind of vaccine platforms do we have, and what can we learn about it, and can we have vaccines developed before the next outbreak comes, for example, Rift Valley fever, Nipah, Marburg, etc. And then coming back to this graphic showing the 8 ways to deliver the genetic code into the cell, and I'm talking now about the viral vector vaccines. So viral vector vaccines were licensed already, so they're kind of new. They're not the traditional approach where they use the virus and attenuate the virus, or use the proteins and develop a vaccines based on the proteins. So the viral vector vaccines are kind of new, but pre-2020, for example, the Ebola VSV-EBOV was approved and licensed by the WHO and EMA and FDA, and the adenoviral vector 26 and the MVA against the filovirus was also licensed in 2020. And with the COVID pandemic, we have new vaccine candidates on viral vector platforms that were licensed, like the ChAdOx from the Oxford University, again an AD26, and also an AD5 from China. So the viral vector vaccines can be based on different viruses. So we can use the VSV, which I talked about before, the adenovirus vector, the MVA, measles, and more. And viral vectors act as a delivery system to invade the cell and insert the code of for a different antigen, for a specific antigen, what you want to tackle. And the viruses that are used as a vector are engineered to carry the target gene, like spike or Ebola glycoprotein. The virus itself is harmless, but the immune response is mounted without the developing of a disease, and the genetic material instructs cells to produce an antigen stimulating an immune response, and some viral vector vaccines enter the cells and cause them to make viral proteins. Some are really replicating, make new viral vector vaccines, and inducing a more stronger immune response. And some, like the VSV-EBOV, carry the antigen on the surface. So on the left side, you can see, for example, an adenoviral vector, where you have the wild type in the top, where you have the different genes encoding, which encodes the adenoviral genome with the inverted terminal repeats left and right, and then you have the early genes and the late genes. And when you want to develop an adenoviral vector vaccine, you need to delete specific genes that help to develop a full replication cycle of the virus. So this adenoviral 5 vector shows you that the early genes are deleted, and you can include different transgenes. So the gene of interest, for example, the glycoprotein or the spike protein. On the right side, you see the VSV, and this is a bit of a different strategy because they don't really delete several genes out of this whole genome. They switch the protein. So VSV has a glycoprotein, and in general, it's kind of switched with a different glycoprotein or spike for the transgene of choice. And I would like to talk about the modified Vaccinia ankara virus, because this is kindly also quite used in African region, here in DRC, for example. The MVA is a vaccine developed against orthopox virus and was used against smallpox. And now with the Mpox outbreaks, MVA is now introduced again as a vaccine candidate against now Mpox. On the other side, you can use MVA as a recombinant vaccine. You have the possibility to introduce a gene of interest into the MVA against coronavirus, against HIV and Zika. And so MVA was also developed here in Germany by Gerd Sutter, and therefore I would like to pause here and to highlight that Gerd Sutter was a really extremely talented, a really great person, virologist, vaccine researcher who passed away last year. So let's then have a look to the diverse platforms. You have on one side the adenovirus-based vectors, recombinant vesicular stomatitis virus, MVA. You can use measles, Newcastle disease virus, cytomegalovirus. And each of the vector have different pros and cons when you think about manufacturing. So some of them are really easy to produce, some are not easy to produce, some are really easy to have a high-scale manufacturing process, some not. Some have a really high immunogenicity induction, some not. And some have a safety concern, some not. So if you want to develop a vaccine against a specific pathogen, you really need to think about what is the target population for which approach. Is it for an outbreak, or is it for preparedness, or is it for children, is it for pregnant women? And then deep dive into all the different signatures of each specific viral vector to choose the best for your disease. So in general, you have on the left side, I wanted to highlight the pros in general for viral vector vaccines. So it's a really well-established technology. In general, they induce strong immune response, and really they involve B and T cell responses, whereas the drawback would be that some of the viral vectors have a previous exposure to the vector, which reduce efficacy and complex manufacturing. So to deep dive into the cons would be pre-existing immunity, and this is really important to highlight. The pre-existing immunity to viral vector vaccines can neutralize viral vectors before it delivers the antigen, lowering vaccine efficacy. So antibodies and T cells prevent the vector from infecting cells and expressing the antigen, and so you don't have a good vaccine response. And this is, for example, for Ad5. Here in some region, it's really the zero prevalence is high, and this was one of the case for the Ad5-based COVID-19 vaccines for CanSino, for example, or Sputnik. They faced similar concerns about reduced efficacy when populations have the Ad5 zero prevalence. But also if you use a homologous prime boost vaccination with the two shots of the same vector, this may impact the boost vaccine response. So addressing the challenge, you can develop alternative vectors like adenovirus vector 26 or chimpanzee adenoviral vectors, and use a heterologous prime boost strategy. The manufacturing, it's easier than some technologies, but require really high advanced technological facilities, require expertise involving the complex process. We need to think about the cell culture, the viral engineering, and how to purify the vaccine out of the cell culture. The growth requires live cell systems, which is a resource intensive, and high-quality bioreactors. So viral vector vaccines are quite expensive to produce, and the viral vectors yields can vary depending on the cell lines, culture condition, and production scales. So to address the challenge is building more knowledge and building global capacity, and also develop modular, scalable production systems in the future. The pro is that viral vectors have a really strong immune response, and viral vectors mimic natural infections, triggering robust immune response. No adjuvant is required. The immunogenicity is a result of the pattern of the viral vector and mechanisms through which it activates the immune system, and they are able to induce long-lasting immune response. And one aspect I would like to highlight is also that we have on the one side replicating versus non-replicating viral vector vaccines. So on the one side, these vaccines are highly efficient in delivering genes and enabling host cells to produce high concentration of antigen. And if you have a replicating vector, this means that it produces new viral particles in cells they infect. So then they can again infect other cells. So there's a really strong immune response, but you have a safety concern sometimes. Or you use a non-replicating vector, which is unable to make new viral particles. And so it may require a higher dose of the vector or a prime boost regimen to elicit really sufficient immunity. And this is an example of an adenoviral vector vaccine and how it integrates into the cell and how it induces a strong immune response. I don't want to go into the details here, but just to highlight that if you have an adenoviral vector, it binds to the receptors such as CAR and CD46. It will trigger immune responses by the TLRs. If it integrates into the cell, this will also be recognized by TLRs, by other mechanisms, and activates inflammatory cytokines, triggers innate immunity. And then you have, on the one side, induction of humoral immunity, production of high affinity neutralizing antibodies, hopefully, but also a strong T-cell response, cytotoxic CD8 T-cell response, and hopefully also a good memory response. So the question is then, how do vaccines really work, and viral vector vaccines in specific? And each vector induces a different immune response. So we can't say viral vectors are doing this and this and this. We need to really see each vector independently. What kind of platform is it? What kind of antigen do we have? What kind of cell types is the vector infecting? What is the entry mechanisms? What kind of TLRs are stimulated? Is it a replication or non-replication viral vector? And how high is the antigen load for after vaccination? And here I would like to highlight that the question, how can we learn more about vaccines? And we need to do clinical trials, and we need to do really smart, thoughtful clinical trials to dissect immune responses to vaccines. So this is a standard trial where you want to investigate into adaptive immunity. So you have a prime boost vaccination, you collect a bit blood, serum, investigate into antibody responses. This is really important, but on the other side, you can do more. You can investigate innate immunity if you go for day 1, 3 after vaccination and really look what is the vaccine doing, what kind of innate immunity is being induced after vaccination, to better understand which mechanisms the viral vector is inducing, to better understand what kind of adaptive immune response is the result. So this is kind of what I did before in my previous work and before CEPI, working at the UKE with Marilyn Addo, and also one of my PhD students, Marie Westkamp, developed kind of a clinical trial collecting a lot of blood samples, and then we wanted to investigate what is the transcriptome after vaccination, what are the cytokines, chemokines, innate cell activation, T-cell responses, so that we really have a comprehensive picture of the vaccine. And this is, I don't want to go into the detail because it's not the topic of today, but just to think about what kind of data you can generate after a vaccination. Of course, you can do the ELISA and neutralizing antibodies here, PNT50, the vaccine-induced antibody responses. But then it's also sometimes important to really understand what kind of subclasses or isotypes are induced after vaccination. Is it really the vaccine response you need for this specific pathogen? And then, for example, also the transcriptome, do we see in red, you can see an induction of several genes. So the vaccine is really inducing a strong innate immune response. And this is also seen here with the cytokines. And it's important to identify immune signatures associated with specific viral vector vaccines or with other platforms to choose later on what is the best vaccine for your target population, for your pathogen you want to protect. And therefore, I would like to highlight the relevance of systems vaccinology, because this can train us about how vaccines work by analyzing innate immunity, and if we really can predict the breadth or magnitude of adaptive immune response when we understand the innate immunity. And there are some papers, and one paper showing here that comparing different vaccines and what kind of profile this vaccine is inducing, if they are inducing NK cells, B cells, monocytes, inflammation, and then collating it with other vaccine candidates to get a deeper understanding of the interaction. And here Bali Polendran is the leading a lot of systems vaccinology approaches. And finally, to also highlight that innate immunity can also be linked to AESIs. So for adverse events of special interest, there are also some approaches like the INSIS. They work on this approach. So to sum up, I'm trying to catch up now a bit. So viral vector vaccines can recapitulate the natural infection process of specific pathogens, and they trigger really the classical acute inflammation and immune detection through the natural production of pathogen-associated molecular patterns. They have a systemic immunity induction, and they induce innate and adaptive immune response really strongly, and T and B cells. They induce immune responses without the complete hazardous of the pathogen, and they don't need an adjuvant. And so there's really good vaccine platform for a robust and long-lasting immunity. So, and with this, I would like to give a brief overview about our 100 days mission. And here, which was published a few weeks ago, there was a research from modeling experts from Imperial College, and they highlighted the impact of the 100 days mission. So if we would have a COVID-19 vaccine within 100 days, and what that would be the result, it would be the result that more than 8 million people who died during the COVID pandemic might be alive if we had accomplished the 100 days mission. So what does it mean? So here in this graphic, it shows you the traditional approach. We need about 5 years at least. So this is the minimum. During the COVID pandemic, we were able to have the 350 days. If we really do compressed timelines, we are able to have 250 to 300 days for the vaccine development. But we want to have the 100 days. And this needs to be, we need to have a paradigm shift. We need to shift a lot of activity to the preparedness. So we need to have a pre-outbreak phase. So this is a shift to preparedness we are working on. And here, for example, the knowledge on viral vector vaccines need to go into the preparedness phase as well. How can we manufacture quickly? What do we know about the immunity, about the safety? And then prepare a scientific toolkit, develop infrastructure, and also policy strategies to be able to respond super rapidly after the outbreak. So 100 days, what will it take? Different strategies need to be implemented. We need the disease surveillance. We need to understand immune response markers. We need to create vaccine libraries against representative pathogens. We need to establish global manufacturing capacity and also getting clinical trial and laboratory networks ready. And here, for example, for viral vector vaccines, they are also in the vaccine library topic. And CEPI has a vaccine portfolio about more than 50 vaccine platforms. And you can see the different active vaccine platforms in the number here below. And for example, for Lassa, we have at the moment 3 active vaccine platforms. And this is a list of all the different platforms showing that CEPI is funding on the one side viral vector platforms, but also proteins and mRNA vaccine candidates, so have a broad range of different platforms. But CEPI is not only supporting vaccines, but also really manufacturing, getting research preparedness available, have projects on enabling science and cross-cutting activities over the world. So to sum up, the viral vector vaccines act as a delivery system. They mimic natural infection, which leads to a really strong and long-lasting immune response. They can induce B and T cell responses. We have a different variety of viral vectors available, and we can choose which one is the best, maybe. Each vector has specific signatures and pros and cons related to manufacturing, safety, and immunogenicity. And the platform knowledge can be critical then for the vaccine development and for decision-making processes. And develop vaccine candidates for priority pathogens will. So here we need to have a more knowledge and increase the network and collaboration, because if we are doing silo work, we will not get ready for the next pandemic. And this is all I have, and thank you for your attendance.
Thank you very much, Christine, for a very enlightening talk. I've done this for 8 webinars now, and I still always learn something new every time. Yeah, thank you very much for that. Quite interactive. I'll open up the forum now for any questions we might have for Christine, and I can see a couple of them already in the chat box, which I will read out. And 2 hands are also open, so I'll be able to quickly just ask one question and then ask Tariq to ask his question. So the first question that has come in is from Dr. Navneet. What, in your view, would be the maximum time consumption during the accelerated development of the candidate vaccine during an outbreak setting, preclinical or clinical?
So, so in an outbreak setting, so if we have a new virus, or if we have, so in our view, we will have already a vaccine available before the outbreak. So we have preclinical data, and preclinical data is then available, and then the outbreak hits, and then we need to be ready immediately. And we have seen that the vaccine production, specifically for mRNA, can be super rapid, a few weeks, going into the animals, showing protection in animals, and then go into phase 1. So a few weeks to a month, I would say. But I'm not sure. Maybe he can clarify this question.
Dr. Navneet, if you can clarify, I can see your hand up. Please go ahead and mute and ask your question. Are you able to unmute? Let me just do that for you. 1 second. Please go ahead and ask your question.
So
Dr. Navneet, can you hear us?
Okay. So maybe I can also highlight that if we think of a disease X, I think it will not be a really new virus. So it will be virus. It will be probably a virus family which we know. We need to have an animal model which we know to. It's not unmuted.
Hello.
There you go.
Okay.
Yeah. Yeah, so my question was particularly, like as you mentioned, it took us 320 4 days, I believe, or 26 days for the development of this COVID vaccine. But in regards to the 100-day mission, I like if we cut down time, if we cut down time, we know that the maximum with the process of reverse vaccinology that was very essential in SARS-CoV-2, we were able to achieve the genomic sequence and get those data as quickly as possible, and the development accelerated as quickly as possible. So my question to you is like, even in the 100-day preparation, is it the clinical development part which is going to take maximum time, or is it the
Yep.
preclinical where we have to measure the toxicity report and those preclinical assessments? So my concern is, uh, where exactly are we going to invest? Is it towards the lab side or towards the clinical academia side?
So I would think that the clinical part need to be focused and will take a lot of time. But in the
Yeah.
100 days mission, we need to prepare for this outbreak. So the question is, what is the next outbreak? Is it really a disease X where we know nothing? So then I think the 100 days mission is difficult to map. If we
Yes.
have the SARS-CoV-3, for example,
Yeah.
this will be more easier because we have probably, when it has the same a similar pathogenesis, we can
Yes,
use the the preclinical models and everything. So really, it depends on which scenario do you have.
so that was my point actually. So to understand, basically, this 100-day CEPI mission is particularly for disease X, or also for the pathogen that are already in the which we had already experienced. And that was my first question. My second question is regarding to cut down this clinical development, which was my first question. Do we have to also approach innovative study design, like adaptive seamless trial designs, or maybe other designs that may cut down, where we can run parallelly all the sites, or maybe we don't have to do phase 3, and regulators can accept the fact that phase 1 2 is giving you a robust data, can go ahead with the pre-qualification.
Yeah, and I think this is key to have really different sites available to run in parallel several clinical trials to collect the data, so that you're fast and not have one step after, so that you have, of course, first phase 1, but maybe you can have a parallel investigation so that you collect important data for accelerating phase 2, accelerating phase 3, and have alternative clinical trial designs.
So, in regards to that,
So,
yeah, so in regards to that, as I'm also working in one of the CEPI's 100-day mission project, my regards is how important, according to you, is a hospital and community-based surveillance to understand background rates before, there are a lot of candidate vaccine, investing on different platforms, for example, at the time of SARS-CoV-2, there were a lot of investors, spending billions of dollars in each and every platform, and I think it's high time that now we clearly define which platform is suitable in that particular setting. If we know or understand the background rates of different diseases in the community, so that we also saw the TTP, which was seen with the AstraZeneca vaccine, which is taking a big issue currently in the market for the safety concern. So, do you think we should be much in regards to preparedness that you mentioned, that we should strengthen the health system capacity for strengthening these hospital and community-based data?
Yeah, and I think so CEPI has also announced 2 programs on this to work on this. So you can reach out to me so I can connect you with the program. So we are working on this as well.
Great, thank you very much. I'll also share the contact in case you need to contact Dr. Christine for more information. We'll go on to the next question from Dennis Lapuente. Thanks, Christine. Great talk. Having mRNA vaccines at hand with good immunogenicity and easy adoption or production, what is the niche for www, better one-shot immunogenicity?
Yeah, so it really depends what I mentioned. What kind of pathogen do we have? Which kind of disease? Do we have an outbreak? I think mRNA have shown that they can be rapidly produced, that they can induce really strong antibody responses. But do they really have long-lasting immunity? Do we have robust memory B-cell responses? This is maybe for SARS-CoV-2 it was the ideal vaccine, but maybe not for other other pathogens. So we need to figure out and identify what kind of viral vector vaccine would be more suitable, for example, for a disease where we need to have a long-lasting immunity. T-cell responses might be key for some of the diseases and not antibody responses, which is more advanced for the mRNAs. They showed really strong neutralizing antibody responses. Or if we want to go to mucosal immunity, our mRNA vaccines are really feasible in inducing mucosal immunity or by using different devices. And so there are so many different questions. And we need to go from pathogen to pathogen, and it's not the time to say we learned from the pandemic that mRNAs are the best, because they have also drawbacks. They have also problems and not useful for all the populations. So therefore, I would go for a broad approach.
Thanks for that. I will ask one more question in the chat and then unmute Tariq for him to ask the question. The next question in the chat is from John Michael, who is asking, as the SARS-CoV-2 virus continues to mutate, how does the viral vector vaccines protect against new variants? And what is the mechanisms that ensure vaccines to neutralize emerging strains?
Can you repeat the last? I didn't.
The last part is, what are the mechanisms that enable vaccines to neutralize emerging strains?
Yeah, so for SARS-CoV-2 is the problem that we still have the viral spread. It still continues to have evaluation of new variants. And in my opinion, the only useful strategy is to develop next generation vaccines, like mucosal vaccines, for example, really to limit the spread, because we don't want to have the viral spread to induce new variants, to have again new adaptation of vaccines. And here viral vector vaccines are maybe on the drawback, because it's a bit more complicated to insert the new antigen to the viral vector compared to the mRNA platform. So my question would be focusing on what kind of vaccines do we have to limit transmission and to stop the transmission, and do we have such kind of vaccines? And if not, we still need to adapt the vaccine candidates for the new strains. And I think at the moment WHO also had announced again new variants which should be used for vaccine developers to be included in their vaccine formulation.
Thank you. Tariq, if you can unmute and ask your question.
Am I audible?
Can hear you.
Can hear you.
Thank you for the talk, Christine. A few of my questions have been answered. Just one more question, which is on the social side. Um, while we tend to think of the fast development of the SARS-CoV-2 mRNA vaccines as a positive, some on the other side ask the question that how come we were able to develop such a vaccine that first? And so the question is, in the context of the 100-day mission, do you factor these kind of social variable where you need to communicate and really pass out the message adequately? How does that work? Is that a variable you can control for? Thank you.
Yeah, this is an important topic, and this is not really one of the focus from CEPI. So we are really focusing more on the technical perspective and safety issues. So we have also programs really trying to understand, for example, TTS or myocarditis to address these concerns in the population. But in general, so what is our communication strategy? We don't have a program at the moment, and this will be a problem if there's a new outbreak coming. But I think it depends also on the pathogen. So if we have really a pathogen which has really a high transmission rate, which is highly pathogenic, people will more easier take the vaccine. So it depends really on the pathogen. What is the next disease? How harmful is the disease? How deadly is the disease? And we need to prepare for everything. And try to be transparent as much as we can, and try to also address the safety concerns and from the population.
Okay. We have a question from Abdus Salam. What are your comments about long COVID? Do the viral vectors vaccines remove this persistence?
I don't know. I don't have any answer to this. So sorry.
Yeah. Okay. And there's a question from Jatsada. How do we avoid pre-existing immunity against viral vectors? Can we use a stealth strategy or formulate the vector with liposome?
Yeah, so the viral vector pre-existing immunity is quite so important. And what I experienced in my research with using viral vector as a vaccine, we haven't seen so much issues on viral vector immunity after prime boost vaccination. So I think when we think about viral vector vaccines, they can be used and we can boost at a specific time with the same viral vector. So we didn't see any strong impact. However, if we have for Ad5, which is really a virus which circulates in the population, really making a disease, really have a strong immune response, then we have a problem for pre-existing immunity, and this needs to be addressed. So, and here I would say, for example, the Ad5 is not the best candidate for when we think about pre-existing immunity, although it's not that there's no immunogenicity issues, there's no vaccine-induced immunity, but it diminished, so it's a bit reduced. However, we can see an effective vaccine, although there's a pre-existing immunity. So we need to be aware, we need to monitor, and we need to develop new strategies like the chimpanzee or the Ad26 or different prime boost strategies, but it should not be stopped because of pre-existing immunity.
Okay, great. There's a question from Charity. Some countries are not very good at keeping track of the vaccines received by an individual in their lifetime, or for whatever reasons. If a person's vaccination history is unknown and they probably have received triple V, will this not compromise the efficacy for future vaccinations?
No, I don't think so. So because I don't think that vector immunity will play an important role after in individuals that receive this kind of platform before. And this has been shown also first. So there are different trials ongoing. So there's an VSV, and also I need to check. So there are different clinical trials at the moment ongoing, testing the impact of viral vector immunity against VSV. Yeah, because of the EBOV. So VSV EBOV, they test against VSV Lassa, for example, is ongoing. So there are some trials ongoing to investigate viral vector immunity, and there's no critical impact at the moment seen, but the data is not complete yet.
Okay, thanks for that. There's an innovation question here from Desmond. Thank you, Christine. Have you heard of a device called Mango, invented to automate viral vaccine manufacturing from start to finish within a short time as fulfillment of CEPI's 100 days mission? What areas with respect to this automated Mango device can regulators look into to ensure safety and quality vaccines for approval?
Yeah, I need to deep dive into this. I can't say this now.
Yeah. Yeah, and that's what I like about this webinars, because then there's quite a lot of cross-sharing of information. So that's something that we will definitely have a look into. Sounds quite interesting. Moses has asked, phase 3 is absolutely required to prove efficacy, either directly through an efficacy endpoint or indirectly through a surrogate marker. Perhaps the question should be about how to make it more efficient in a manner that is acceptable to regulatory authorities. I think that's a comment. I don't see a question in there.
I agree.
Yeah. Yeah. And Eugene has asked a question to the Kenyan team, which I think I will throw to my colleague Makobu, who is here. And the question he has asked is hopefully to pose to the panelists: how ready is Kenya, or how positioned is our country in terms of stakeholder investment to allow commercial production of viral vectors to produce, for instance, mRNA vaccines? If my colleague is still on the call, he could take that. Kobo, are you still here? Seems you scared him off. Let me just see whether he's still there. Okay. So far, what we're doing as a country, yes, we are actually working towards that. And one of the things that we're doing is exactly something like this, where we've also identified that we have a lot of fragmented personnel with different skills across the continuum, either within the labs, within regulatory, within manufacturing. And what we've done is try to bring all this skill set together to first identify what the baseline is for the country and also within the African continent. So there are training blocks within East Africa, within West Africa, within South Africa that are also running the similar thing as what we're running right now within the East African bloc, to just build that workforce capacity, to have African scientists capacity build on vaccine research, development, and manufacturing, with the hope that within this period of time, we'll build enough capacity to be able to set up institutions that are able to manufacture these vaccines within the continent, that ends up now serving a wider population. So yes, the interest is there. The work has already begun. And this is why we're having such platforms such as this webinars, the vaccinology courses that we offer, which is a 2-week course, and the apprenticeship for skill sharing with manufacturing institutions, where we place apprenticeships for a period of 3 months to 12 months to just be able to understand that skill set and transfer it back to their respective countries. We just concluded a 2-week course that was in the month of May this year, where we had over 19 countries represented, where they had sent in representative apprentices just to learn and to also share that skill set from the point of public health and distribution of the EPI programs all the way to lab research and all the clinical trials. So the work has begun. It's an interesting period we are in where we've learned a lot from the COVID period, and country as part of the African continent has stepped up. I think that's what I can say about that. And Desmond has shared about the Mango device. Thank you very much for that. We'll take one more question from Fred. Are there pharmacovigilance around prospective risk factors of AstraZeneca vaccines with progressive effects and mortality being seen?
So I'm not sure about AstraZeneca, I think so, but CEPI is funding pharmacovigilance projects for their vaccine developers. So this is part of our programs, and we continue to monitor this in our trials.
I do not see any other questions in the chat box. Maybe I can give Christine an opportunity for last comments before we close down. And I'm also posting post-webinar feedback form on the group. The slides will be available within our YouTube page, which we'll be able to circulate. If you're able to fill in the form that I have just posted, we'll be able to get your email details. And if you have any questions for our speaker, then we'll also be able to get that information based on the form that I've just posted on the chat box, and share the link for the YouTube page for you to be able to review this webinar and also share it with your colleague. Last comments from you, Christine?
Yeah, I don't have really a comment. So just thank you for being here. Thank you for the questions, and looking forward for future discussions. And reach out to me, and happy to discuss.
Okay. Thank you very much, guys. We are almost at the end of this webinar with 15 minutes to spare. This was quite an interactive session towards the Christmas holidays. Thank you very much for attending, and we hope that we will see you in the new year. Looking forward to the next batch of webinars that we will be running in the year 2025, all focused within the vaccinology research, development, and manufacture space. From me to you, it's thank you very much and happy holidays. Have a good day. Bye-bye.
Holidays. Thank you, Caroline. Thank you all.